Welcome to our 5th Friday Flip Day. We'll be meeting in the STEM foyer as usual to do some super active metabolism drawing. Now, this is going to be an exercise that is going to really get you ready for the second exam. And my hope is that this will be the first time of many that you will sit down with these pathways and draw and redraw and redraw. And in fact, that is why I have thoughtfully and purposefully scheduled your second exam for March the 21st, the Tuesday after spring break. This is because I want you to take your metabolism poster home with you or on whatever vacation you're going on. You can see some of my prior students here have done so, taken it to their family festivals, taken it to study with their dog, and spend time drawing and redrawing those pathways and learning them a little bit each day. Spend 20 minutes a day. And if you do that, I promise you, in fact, we know that that leads to a more effective, deep learning of the material. So your challenge, should you choose to accept it, is to be ready for an exam right after spring break. And not just that, but to be rested and vibrant and ready to show me what you know. And my hope is that with this approach, we can have the first ever exam on which everyone receives a passing grade. I don't know what I would do if that happened. I think I would have an in-class festival, or I would cut a mohawk. No, probably not. But you know, you never know. I would do something crazy if you all could achieve that. And as you're working today to get ready to go on spring break, please remember to finish off your edited hypotheses. I will make certain to give you final grades on those if you can get those in. And I don't want you to think about them over spring break. I just want you to think about having fun. And one of the funnest things is constructing new knowledge while you're traveling to crazy awesome places. So remember, when we had left off last time, we had been talking about metabolism, and we had been specifically talking about aerobic respiration. However, we had made note that not all organisms respire aerobically. In fact, we had talked specifically about the flexibility of E. coli and the fact that when there's no oxygen, it'll simply turn to nitrate as its terminal electron acceptor. So that's where we're going to dive in today, looking at something that is called anaerobic respiration, meaning that something besides oxygen serves as the final leg of the relay of the ETC. Instead of O2 at the end of their ETC, they have something else. For example, they might instead use something that is a highly oxidized form of nitrogen or carbon, or even an oxidized iron. So things like nitrate, things like even carbonate, things like iron 3+, the ferric form of iron. So these highly oxidized molecules, sometimes even CO2, can be utilized as terminal electron acceptors. We know that they're not as good as oxygen, and by good, I mean they don't have as high of a standard reduction potential. However, they are still fairly highly oxidized, so they have a reasonably high affinity for electrons. E. coli, we mentioned this already, can utilize anaerobic respiration, can and does utilize anaerobic respiration. So it utilizes nitrate as its terminal electron acceptor when it is respiring anaerobically. Here is a balanced equation showing nitrate, NO3-, going to nitrite, NO2-, which, hello, toxic, super toxic. So that nitrite must have to be converted into something else quite right away. But this is possibly one way that E. coli could continue to generate energy even in the absence of oxygen. Interestingly enough, this is also considered a nitrogen incorporation reaction, enabling E. coli to now use that nitrogen in some way to build macromolecules, say amino acids and the proteins that they comprise. So some anaerobes use sulfate. And this is where things get really cool. So the use of sulfate as a terminal electron acceptor is something that we are actually seeing happening right now in the lab. So remember your Winogradsky column? this is the Team USA Winogradsky column, and I'm not quite sure who's in that group. Don't tell environmental health and safety that this isn't fully labeled with the names of everyone. But in any case, notice that we see some interesting bands of growth in this Winogradsky column, really good looking. But one would recognize that organisms that respire aerobically would have to live up in the top of the Winogradsky column. And then down in the bottom, there would be two strategies of life that would be available to these bacteria. One would be obviously anaerobic respiration. So sulfate often serves as a terminal electron acceptor down in these regions. And when sulfate is reduced, of course, lots of bonds to oxygen when we're talking about sulfate. If that gets fully and completely reduced, the product is hydrogen sulfide gas, the really smelly rotten egg stuff. So this hydrogen sulfide gas is wafted up through that Winogradsky column, and interestingly enough, serves as an electron donor for anoxygenic phototrophs. Cool, right? One man's trash is another man's treasure. So other anaerobes such as methanogens, which Mackenzie and Malin are going to tell us that anaerobes such as methanogens live in the rumen, and I'm going to come back to that here momentarily. So they use CO2 as their terminal electron acceptor, and check that out. If CO2 gets fully reduced, we get CH4, methane gas, methanogens. Yeah, they give off methane gas, which is why if you've ever seen a manure pile burst into spontaneous flames, that's why methane gas is highly flammable. So anaerobic respiration, as we all know, is going to generate less energy than aerobic respiration. Clearly, that is going to be the case because none of these oxidized molecules have a standard reduction potential of 0.82 the way that oxygen does. The thing that comes the closest is nitrate, 0.42, but still, that is not near as high. So less energy is generated in the transfer of electrons throughout an electron transport chain that ends in nitrate than would be throughout an electron transport chain that ends in oxygen. Sulfate is even less energy generated, and CO2 even less. So it's interesting to think about that. I mean, why respire anaerobically? Why would any organism do that? Well, we know there's a lot of answers to that question evolutionarily. But if you just think about environmental niches, there's probably not a whole lot of competition out there for using CO2 as your terminal electron acceptor. So despite the fact that per CO2 molecule, there's not as much energy generated, or per oxidized substrate, there's not as much energy generated. However, there's not a whole lot of competition either. So it allows these organisms to take up rare environmental niches and be the king of their castle in those sort of niches. So I hope that helps you understand that respiration isn't always aerobic. And one of the areas that is really confusing, and I'm going to state and restate this because this tends to be very confusing, is that anaerobic respiration is not fermentation, right? Remember, in fermentation, it means that the whole ETC has quit, the whole TCA has quit, the whole transition step has quit, and the only thing running is glycolysis. The only thing that's similar in anaerobic respiration and fermentation is that they take place in the absence of oxygen. However, anaerobic respiration still involves respiration. It still involves an ETC. It still involves a terminal electron acceptor. So as we answer this next question, that's something important to keep in mind. E. coli can live using aerobic or anaerobic respiration. But what strategy does this bacterium use to live in the absence of any externally derived electron acceptors? That's when we can fervently answer fermentation. That's when it has to use pyruvate or a derivative of pyruvate as an internal electron acceptor to keep glycolysis going so that it can just scratch together those two ATPs from glycolysis. Beautiful. Fermentation occurs in the absence of an externally derived electron acceptor. Whether that acceptor be oxygen, whether that acceptor be nitrate, doesn't matter. Fermentation doesn't have an externally derived electron acceptor. We return now to our poster. One of the things that I want to quickly make note of and we did briefly talk about this when we began our map-looking journey into metabolism, that is that glucose is by no means the only sugar that can enter into glycolysis. And in fact, we could look at other sugars such as galactose, another monosaccharide, and in 5 different steps, this can be converted into glucose 6-phosphate and enter throughout the rest of the cycle. Additionally, we know that things like mannose, another monosaccharide, can enter as fructose 6-phosphate. So we recognize that there are other entry points into metabolism. And with the exception of a kind of sneaky entry point that fructose sometimes uses, for the most part, they all enter prior to the first committed step. So this is key. That's actually why we call step 3 in glycolysis the first committed step, is because it's the step at which all sugars have entered. And so it's the first point of shared regulation. And that is why it gets that name of first committed step. Beautiful. Remember that too, there are disaccharides. For example, sucrose could get broken down into fructose. Fructose could get broken down sucrose could get broken down into fructose and glucose, and then fructose can enter simultaneously with glucose. So we can see disaccharides also getting into catabolism, and of course polysaccharides. So I want to take a look at the digestion of things other than just plain glucose. Looking most fervently at the degradation of starches, and this you can see is amylopectin. It's got a 1,6 glycosidic linkage. It's also got lots of 1,4 glycosidic linkages. So a bacterium that can degrade this sort of starch has to secrete enzymes that will degrade the 1,6 linkage and the 1,4 linkage. So this is interesting because, as it turns out, not every organism can secrete those enzymes. So we actually can assay for those organisms that can do this. We can actually look to see if they have the ability to make these 2 enzymes. So this is a picture of the starch hydrolysis test, and you can see that this particular bacterium secretes alpha amylase and oligo 1,6 glucosidase. So you can see that it has broken down the starches in the region just surrounding the growth there. And so this is really a cool assay because the iodine binds to the starches in the plate. And so if the starches are still present, everything is black because the iodine binds to it. But if the starches are consumed and the bonds are broken and they're broken down, then there's no more starch to which the iodine can bind. And so we get this really good looking halo that surrounds it. So this would be an example of a positive starch hydrolysis test. And this might be used if you were trying to identify gram-positive bacteria such as Bacillus subtilis. Bacillus subtilis is capable of degrading starches, whereas other gram-positive rods are not capable of doing that. So it'd be a cool way to differentiate it from other gram-positive rods. Now these enzymes are very fittingly named oligo 1,6 glucosidase. They break down the bonds between the glucose residues, and more namely, the 1,6 glycosidic linkage. The 1,4 breaking enzyme is slightly less fittingly named, at least in its common name, which is alpha amylase. Alpha amylase cuts that glycosidic linkage. So it's really awesome to relate this to what's going on within the rumen of cattle or other ruminants, say sheep. When we're thinking about the fact that there's this 40-gallon fermentation vat within a ruminant, a cow, a full-grown, cow will have a huge fermentation vat. So when we recognize that there's a lot of fermentative processes going on there and a lot of degradation, not necessarily of amylopectin, but of cellulose and other indigestible starches, there's a whole lot of secretion of enzymes by these bacteria to break down the sugars in that indigestible starch, the polymer of, say, cellulose, into acid byproducts. So remember, fermentation, one product of that is acid. And in the rumen, we actually see the building up of a lot of butyric acid, acetic acid, propionic acid as byproducts of fermentation. And then those serve as sources of energy for the cattle. So here's a great image of one such example of a ruminant. And of course, recognizing that the bacteria are digesting the insoluble starches such as cellulose, in the case of cellulose, cellulases, and releasing the glucose subunits and forming those acidic byproducts. And you know what's really awesome, and you all are going to love this, and Mackenzie and our other ruminant lovers out there, there are on the order of 10 billion bacteria per gram, or let's say microorganisms, because they're kind of mixed. There's everything ranging from bacteria to archaea to protists to fungi, but 10 billion of them per gram of wet weight within the rumen. So it's extremely packed with action and fermentation, a lot going on in there. So we know that the glucose subunits are released when the polysaccharide is degraded, and of course those can enter into glycolysis and be degraded in the ways that we've talked about. So it's cool to think that not only can bacteria degrade these things that certainly we don't see as viable energy sources such as cellulose, but also bacteria can degrade things that we really wouldn't see as donuts. So I think the other day, I can't remember who it was now, came and asked me about unknown number 140, Pseudomonas Aeruginosa. Incredible bacterium that is capable of degrading upwards of 80 some odd organic compounds, and many of them are recalcitrant, meaning that they're compounds that are tough to degrade. So these are superheroes when it comes to bioremediation. They can eat plastics out of landfills and things like that. But we also see that there are other recalcitrant compounds such as TNT, or explosive or oil-based compounds that are very hard to degrade, and that they can take those and convert those into normal metabolic intermediates. Superheroes, right? As we turn our attention to the degradation of fats, the catabolism of fats, we're going to be challenging ourselves to apply all of our new knowledge. So let's go ahead and take our attention to this particular molecule. This is a triacylglycerol. We remember them well from our study of macromolecules. When a triacylglycerol is broken down by a bacterium, much like starches, the bacteria have to secrete the enzymes that will break down the ester linkages between the glycerol backbone and the fatty acids. And of course, those are none other than lipases. So lipases are going to degrade the bonds in between the fatty acids and the glycerol backbone. Then, of course, that leaves this long hydrocarbon chain. And we need to think quantitatively about that hydrocarbon chain and the way it is degraded. First, let's just make reference to the 3-carbon glycerol backbone that is left behind. And we can think about where in metabolism we see a 3-carbon molecule for the first time and give a guess that that's probably pretty likely to be the place where the glycerol backbone will enter into the catabolic processes. So to do that, let's grab Stan's poster and see if we can check out a possible place where we might see a 3-carbon molecule enter into catabolism. So this is going to have to zoom up into the cytosol, into glycolysis, and check it out, where we see the breakdown of fructose 1,6-bisphosphate into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. That's the first time we see 2 3-carbon sugars, or 2 3-carbon molecules made from that 1 6-carbon sugar. So that is the location at which the glycerol backbone enters as dihydroxyacetone phosphate or glyceraldehyde 3-phosphate. Remember, they're in equilibrium with one another, so it matters not which one we say it is because they equilibrate. But glyceraldehyde 3-phosphate and intermediate in glycolysis. The interesting thing about this is that it's tempting to then think that that 3-carbon molecule is always degraded from there, but Jake has probably got his thinking cap on, and he's realizing that if a cell is eating fat entirely, it probably doesn't have a whole lot of glucose. So in fact, rather than using the glyceraldehyde 3-phosphate and breaking it down in glycolysis, a lot of times what it's doing is the reverse pathway of glycolysis called gluconeogenesis, and it's actually using the carbon-glycerol backbone as a precursor metabolite, therefore exemplifying the amphibolic nature, in fact, of the glycolytic pathway in a whole new way. So it's interesting that for our calculations, we won't take into account the 3-carbon-glycerol backbone when we calculate the yield from a fat. Instead, we'll just go ahead and look at the fatty acid side chains. Now, this is where things get interesting. Remember that they're broken down 2 carbons at a time, and they enter into catabolism at the top, and Hannah's yelling at me, at the top of the TCA cycle, right? So we know that they're going to come in over here in the mitochondrion, and they are going to enter in right at the top of the TCA cycle as acetyl-CoA. So that's going to be their site, their entry port, their site of entry. So 2 carbons at a time from the fatty acid chain. So that means we have to understand how many carbons are in a fatty acid chain to determine how much ATP or how many protons can be pumped from the carbons of the hydrocarbon chain or the fatty acid side chain. So I am going to give you an extra credit challenge. So listen up carefully. Here on this triacylglycerol, we have 3 fatty acids. I want to know first, after cleavage of this fatty acid, how many acetyl groups can form. So we've got 3 of these fatty acids. How many acetyl groups in total? So let's just take a look at that. Notice that if we count, there are 14 carbons in the fatty acid side chain. I'm actually going to go through this with you because this has unendingly been a source of confusion for many people. We're going to just go ahead and count the number of carbons so that for future problems like this, you can think through them. This is carbon number 1. This vertex is carbon 2, 3, 4, 5, 6, 7, 8, there, 9, 10, 11, 12, 13, and finally 14. This is a 14 carbon fatty acid. Now think about that. How many acetyl groups would there be from that fatty acid? And therefore, how many acetyl groups in total from all 3 fatty acids? And then I want you to tell me in this extra credit opportunity, how many protons can be pumped. So how many H plus can be pumped from the complete oxidation of these 3 fatty acid side chains? So while proteins are not the cell's more preferred source of energy, they are a possible source. And we recognize that those polypeptide chains comprised of amino acids can, of course, be broken down by enzymes that break the peptide bond, or proteases or peptidases. And when we liberate an individual amino acid, for example, right here, if we assume that this little blue ball was in fact in full structure this amino acid here, which I hope everybody recognizes as glutamate. Glutamate can actually be deaminated. And this, right, Milo, is the loss of this amino group, leaving the carbon skeletons. That's the toxic portion of the amino acid that has to be cleared by the urea cycle. But then, of course, now this skeleton can enter into the TCA cycle as alpha-ketoglutarate. So this is one example of where proteins can get incorporated into catabolism. It's interesting to think about the amphibolic nature of the TCA2, though, and the fact that this reaction could go the opposite way. the alpha ketoglutarate intermediate could be used as a precursor metabolite to make glutamate. So this could go the opposite way as well. So this gives us a down and dirty sort of version of the incorporations of proteins into catabolism. And it also gives us a little highlight on how we might think about them in the context of anabolism as well, like using intermediates from the TCA to make proteins. So that being said, this is largely our final coverage of chemoorganoheterotrophs. Look forward to some coverage now regarding chemolithoautotrophs, rock eaters that utilize inorganic chemicals as their source of energy. And then we'll be looking at photoautotrophs, those light-using primary producers. I have to admit that this is one of my very favorite things to look at the rock eaters. So let's take a moment to do that. And as we're jumping in, I want to show you something. This is actually a drawing that was made by one of my prior students. His name was Cameron, and I actually believe that he is in graduate school right now. But this is one of my favorite pictures. Chemolithoautotrophs rock. I hope at least some of you recognize this as the rock eater from the Never-Ending Story. If you have never seen the Never-Ending Story, that is your homework assignment. Yes, it is your homework assignment to watch that movie. It is BA. So watch the Never-Ending Story. But this is a character that eats rocks. In actuality, in our world, the only organisms that eat rocks are prokaryotic organisms that can utilize inorganic sources of energy. So imagine being an organism that literally eats hydrogen gas for dinner. You see donuts as your source of energy, or hydrogen gas as your source of donuts. Things like hydrogen sulfide, the smelly rotten egg gas that's also toxic in high concentrations, nitrite, these incredibly toxic molecules that we wouldn't dream of chowing down on. Some bacteria love them. And isn't that a beautiful thing? So the hydrogen bacteria, they rip and strip their high energy electrons off of hydrogen. They're like, woo, that's my donuts. Rip and strip the electrons off of those puppies. Sulfur, some bacteria utilize, as I said, hydrogen sulfide. We get iron bacteria that will literally oxidize the ferrous form of iron into the ferric form. So, wow, think of that. They're eating iron. There are some bacteria that are actually both, that can actually utilize both iron and sulfur as their source of energy. And in a minute, I'm going to tell you about my favorite one of those. There are the nitrifying bacteria, which, for example, will chow down on toxic nitrite or ammonia. We see them playing a huge role in the nitrogen cycle. Nitrosomonas, Nitrobacter, huge contributors to the nitrogen cycle, and both are nitrifying bacteria. So recognize that these bacteria are not that different from us, except for that what they oxidize is different. What they then do is pretty dang similar. They rip and strip high energy electrons off of their donuts. Those go to the ETC. They're used to generate a PMF, and there's some terminal electron acceptor. Often it's oxygen. So they're really not all that different than we are. But they certainly thrive in unique niches. They often are in these extreme environments, maybe acidic, where they find their sources of energy. And I want to take you on a journey to my hometown of Leadville, where we can meet one of these epic chemolithoautotrophs, Thiobacillus ferrooxidans. It is a big contributor to acid mine drainage. So at pHs of less than 3 is where it makes itself home. And here is the reaction that we can look at that it catalyzes. Iron sulfide, recognize that? Okay, some of you probably do recognize this. This is actually fool's gold. So when I was a little girl growing up in Leadville, my favorite doorstop was a huge chunk of fool's gold. Iron pyrite is a very common and very, it's a very well celebrated thing in Leadville, Colorado. So some of you from mining towns may have something similar to that. Plus 14 water, plus 15 O2 yields 4 iron hydroxide plus 8. And hopefully you're kind of recognizing what's going on here. Notice the ripping and stripping of high energy electrons. These were highly reduced molecules. That is the ferrous form of iron, the reduced form of sulfur in the sulfide form. So this bacterium is actually ripping and stripping high energy electrons off of both iron and sulfur. It's both an iron oxidizing and sulfur oxidizing bacterium, creating an oxidized form of iron and an oxidized form of sulfur. But I hope some of you are laughing and recognizing what this is. Maybe once or twice you spilled it on your bench top, or maybe that's just me that spilled for sulfuric acid. Oh my gosh, it smokes and everything. So sulfuric acid, a very strong acid, you can see why this is responsible for acid mine drainage. So in my hometown of Leadville, I'm going to show you some pictures, and hopefully your lab TA showed you these, and you saw them on the vodcast for lab a little bit, where we can see the incredible orange color of acid mine drainage. The reason for this is that there are a lot of metal ions that are soluble in sulfuric acid that are not otherwise soluble, and so they get leached into the water, and it turns it really, really orange. I don't know if any of you saw the spill that happened outside of Durango, like in the Silverton area this last summer, where there was a bunch of acid mine drainage that got washed down through the city of Durango. It was crazy, and it was all orange in color, and this is incredibly deleterious stuff. So this is where we sampled for the acidophile that you worked with in lab, so not condoning that activity as a general practice. But chemolithoautotrophs most certainly do rock.